Welcome to a deep dive into the world of viruses. You could call them the ultimate molecular hitchhikers. They're just staggeringly successful, yet they exist in this place that most biologists describe as a shady area between life forms and chemicals. They're living what you might call a kind of borrowed life.
And that borrowed life idea it's everything in virology. So today, we're going to try and cut through that complexity. We'll map out how these particles are built, the really sophisticated ways they hijack our own cells, and, well, why they're still such formidable and constantly emerging threats.
Okay, let's unpack this. I think a great place to start is with the scientific detective story of how we even found them. For decades, scientists knew something was out there causing disease, but they just couldn't see it. It all kicks off back in 1883 with Adolf Mayer, a German scientist. He was studying tobacco mosaic disease, or TMV.
Right. And he found he could transmit the disease just by rubbing sap from a sick plant onto a healthy one. His immediate thought was, okay, it must be caused by some kind of really, really small bacteria.
A totally logical assumption.
It was. But it didn't hold up. About 10 years later, Dmitry Ivanovsky in Russia filters that same sap through a filter that was specifically designed to trap all known bacteria.
And it still caused the disease.
The filtered liquid was still infectious. Ivanovsky actually thought his filter was just bad. But then Martinus Beijerinck, a Dutch botanist, proved that whatever this infectious agent was, it could only replicate inside a living host plant.
Ah, and that's the key, isn't it? He realized this wasn't just some tiny chemical or a toxin. It was a replicating particle, something much, much smaller than a cell.
Precisely. And the whole concept was confirmed in this just stunning way in 1935. Wendell Stanley actually managed to crystallize the infectious particle, TMV.
Wow. Crystallize it.
Yeah, and that sent shockwaves through biology because cells, even the simplest cells, cannot form regular complex crystals. It proved this pathogen was fundamentally non-cellular. It was a package of chemicals, not an organism in the traditional sense.
Which sets the stage perfectly for their structure. I mean, when you look at how these things are built, they are just incredibly streamlined.
Oh, they're minimalist geniuses. At its absolute core, a virus is just a nucleic acid genome enclosed in a protein coat. They're microscopic, most are only around 20 nanometers across, way smaller than a ribosome.
Though we now know some giant ones exist, right?
We do, and it's surprising. Things like Pandora virus, which can get up to 1.5 micrometers, almost the size of a small bacterium.
And that genome you mentioned, that could be pretty much anything.
It's incredibly diverse. The genome can be double-stranded DNA, single-stranded DNA, double-stranded RNA, or a single-stranded RNA. All of that genetic material is packaged inside a protective shell we call the capsid, and that capsid is made of repeating protein subunits called capsomeres.
And we see that architecture everywhere. You have the really rigid, rod-like shape of tobacco mosaic virus. That's a helical structure.
And then you get the complex 20-sided geometric shape of something like an adenovirus. That's called icosahedral.
The complexity really spikes, though, when you look at bacteriophages, the viruses that infect bacteria.
Oh, absolutely. Take the T4 phage. It's often described as looking like a tiny lunar lander. It has this elongated icosahedral head where the DNA is kept, attached to a complex protein tail piece, and even has tail fibers designed to lock onto and inject its DNA into a bacterial cell.
And on top of all that, many animal viruses add a final layer of, well, disguise, the viral envelope.
Right, and that's a really critical accessory, especially for viruses like, say, influenza or herpes. The envelope is this membranous layer wrapped around the capsid, and it's stolen from the host cell's own plasma membrane when the virus leaves.
So it's a disguise.
A perfect disguise. It's got host phospholipins, host proteins, and this is the crucial part, it's studded with these specialized viral glycoproteins.
And those are the keys.
Those are the keys. They're what the virus uses to recognize and unlock the specific receptors on the next host cell.
Okay, so we've got the architecture down. Let's talk about the engine of the virus, replication. This is where we get that term obligate intracellular parasites. They are completely dependent on the host.
They're brilliant cellular pirates. All they bring are the instructions, the genome, maybe a couple of key enzymes, but the host cell has to provide all the labor. The nucleotides, the ribosomes to make proteins, the ATP for energy, All of it.
And that ability to hijack a cell is determined by the host range, right? It's that handshake fit between the virus's surface proteins and the specific receptors on a host cell.
Exactly. And that specificity is why, say, a common cold virus only targets cells in your upper respiratory tract, while a virus like HIV specifically targets receptors found only on certain immune cells. It's very high stakes, very specific lock and key system.
So if we walk through a basic replicative strategy, let's say for a DNA virus, it starts with attachment, then uncoding, where the viral genome gets released into the cell. Then what?
So once that DNA is free, the host cell's own enzymes start replicating the viral genome and transcribing it into viral mRNA. The host's ribosomes, totally unaware they're building sabotage materials, just start churning out the capsid proteins. And then finally, all these newly made parts spontaneously self-assemble into hundreds of new viruses, which then have to get out of the cell.
And we see two really different strategies for this play out with bacteriophages. First, there's the scorched earth policy of the lytic cycle.
That is the virulent strategy. Phages like T4 use this. It's rapid, it's overwhelming, and it ends in the violent destruction or lysis of the host cell. The whole process, attachment, DNA injection, synthesis, assembly, and then this massive release of maybe 100 to 200 new phages. It can wipe out a whole bacterial population in just hours.
But then you have the much more subtle, covert operation, the lysogenic cycle.
Right, this is for the temperate phages, like lambda phage. Instead of this destructive takeover, the phage DNA integrates itself right into the host chromosome. At that point, we call it a prophage.
And it just sits there.
It just sits there, largely silent. Every time the bacterium divides, it passively copies the viral DNA right along with its own, passing the infection down through generations without doing any harm.
That seems like a brilliant way to spread long term. But what's the actual significance of a bacterium carrying around that prophage DNA?
It's medically huge. Just expressing a few of those integrated prophage genes can change the host's entire nature, its phenotype.
So it changes what the bacteria does.
Exactly. This is how a harmless bacterium can become toxic. For instance, the bacteria that cause diphtheria or scarlet fever are only dangerous to us because they carry specific prophage genes that tell them to produce powerful toxins. The virus literally weaponizes the bacterium.
So bacteria are under constant attack. Do they have any way to fight back?
Oh, absolutely. This evolutionary arms race has led to the really sophisticated CRISPR-Cas system. It's basically the bacteria's adaptive immune system.
CRISPR, everyone's heard of it for gene editing, but this is its natural function.
This is where it comes from. If a bacterium survives a phage attack, it will chop up the phage's DNA and integrate little fragments, we call them spacer DNA, into a special part of its own genome called the CRISPR locus.
It's keeping a record.
It's a genetic memory. If that same type of phage attacks again, the CRISPR region is transcribed into RNA. That RNA then guides a Cas protein, like Cas9, right to the invading phage DNA, and it just cuts it to pieces. It's targeted, acquired immunity.
Okay, let's switch gears to animal viruses because they often play by different rules, especially those enveloped RNA viruses like influenza.
Right. With an enveloped animal virus, the whole particle usually gets into the cell, often by its envelope fusing with the host membrane. Once the viral RNA is uncoated, the host cell's machinery starts making viral parts. The cell's ER and Golgi process the viral glycoproteins, which then get shipped out and embedded in the host's plasma membrane.
Ah, so it's setting up the exit ahead of time. And as the new virus leaves, it steals that piece of membrane with the glycoproteins.
Precisely. The new capsid assembles, moves to the surface, and exits by budding. It wraps itself in a piece of the host membrane as it goes. And this is an important distinction. Budding doesn't necessarily kill the cell right away. The cell can survive and just keep churning out more viruses, leading to a more chronic infection.
And then we get to the ultimate genetic saboteurs, the retroviruses, with HIV being the classic example. These are class 6 RNA viruses famous for, well, for reversing the central dogma of biology.
They're named retroviruses because they work backward. They carry their own special enzyme called reverse transcriptase. This enzyme takes their viral RNA genome and uses it as a template to build a DNA copy.
So it goes from RNA back to DNA, and where does that new viral DNA go?
It gets into the nucleus and integrates itself right into the host cell's chromosome, becoming what we call a provirus.
Okay, a provirus. How is that different from a prophage?
This is a vital distinction. A provirus never leaves the host genome. It becomes a permanent genetic resident. The host cell then treats this provirus just like any other gene, constantly transcribing it to make new viral RNA both for new genomes and for the mRNA needed to make more viral proteins.
Which makes treating them so incredibly difficult. You're not just fighting a virus, you're fighting a permanent change to the host's own DNA.
Exactly. That cell is now permanently programmed to manufacture the virus.
So let's shift to the real-world consequences.
Mhm.
Viral diseases, the symptoms we feel, the fever, the aches. It's not always the virus killing cells directly, is it?
No, often what you're feeling is your own body's immune response trying to fight the infection. And the type of damage really dictates the outcome. If a virus hits a tissue that can regenerate easily, like the lining of your respiratory tract, you recover.
But if it hits something that can't,
If it targets cells that don't regenerate, like the nerve cells targeted by poliovirus, the damage can be permanent.
so given all this, our primary defense strategy has to be vaccines.
They're our most powerful tool, by far. Vaccines introduce a harmless piece of the pathogen, maybe just a surface protein, that stimulates your immune system to build a long-term defense. It's immunological memory. It's how we completely eradicated smallpox.
And since antibiotics are useless against viruses, we need very specific antiviral drugs.
Right, and antivirals have to be clever. They're designed to block steps in the viral life cycle that are unique to the virus. So, for example, acyclovir inhibits the specific DNA polymerase that the herpes virus needs. AZT, for HIV, blocks reverse transcriptase. And newer drugs like Tamiflu block the enzyme influenza needs to detach from the cell.
The viruses that really keep public health officials up at night are the emerging viruses, the ones that seem to come out of nowhere. The source material points to three main reasons this happens.
The first one is just simple mutation. It's especially a problem in RNA viruses because their replication enzymes don't have a proofreading function. They make a lot of mistakes.
A lot of typos.
A ton. And a few random mutations can be all it takes to change a virus's surface proteins, just enough to let it jump to a new species.
The second factor is the spread from previously isolated populations. The source points out how HIV likely existed for decades without anyone noticing. But then, changes in human behavior, affordable international travel, for instance, allowed a rare virus to trigger a global pandemic.
And the third cause is the big one. It accounts for about 3/4 of all new human diseases. They spread from other animals. The animal acts as a natural reservoir.
Like HIV from chimpanzees or influenza from birds and pigs.
Exactly. And the threat of something like the H5N1 avian flu is a perfect example of a virus that has a huge reservoir in birds, just waiting for the right mutation to jump effectively to humans.
And type A influenza gives us this perfect, terrifying example of how all this can combine. Its genome is made of 9 separate segments of RNA, which allows for something called reassortment.
This is what makes flu so dangerous. If a host animal, like a pig, gets infected with multiple strains at once, say a human strain, a bird strain, and a pig strain, those 9 RNA segments can all get mixed and matched when new viruses are being assembled.
A molecular shuffle.
A total shuffle. And you can end up with a brand new virus with a combination of surface proteins your immune system has never seen before. That's exactly how the 2009 H1N1 swine flu pandemic started.
Before we wrap up the pathogens, let's just briefly touch on plant viruses. How do they get around?
They have 2 main routes. There's horizontal transmission, which is from an external source, maybe an insect bite or damage from the wind. And then there's vertical transmission, where it's inherited from the parent plant through seeds. Once inside, they spread from cell to cell by forcing open the little cytoplasmic connections between plant cells, which are called plasmodesmata.
Okay. Finally, we have to talk about the strangest thing in the source material. Prions, a pathogen with no genetic material at all. It's just an infectious protein.
The discovery of prions really turned biology on its head. These things cause these horrible degenerative brain diseases like mad cow disease and Creutzfeldt-Jakob disease.
So what is the mechanism? How can a protein be infectious?
It's a chillingly simple idea. A prion is just a misfolded version of a normal protein that's already in your brain. When this misfolded prion bumps into a correctly folded normal version of that same protein, it acts like a template. It physically forces the normal protein to misfold into the prion shape.
And then those two find 2 more. A chain reaction.
A chain reaction. These misfolded proteins then aggregate into clumps, which causes cells to malfunction and die.
And what makes them so profoundly dangerous?
Well, for one, they act incredibly slowly. The incubation periods can be more than 10 years, which makes them almost impossible to track. But critically, they are just about indestructible. Normal cooking temperatures, sterilization methods, they don't deactivate them. There's a biological hazard we still haven't really solved.
Wow. So, what does this all mean? We've gone from the simple elegance of a capsid to the molecular hijack of the lytic cycle, the covert operations of the lysogenic cycle, and now to these terrifying infectious proteins.
The whole field just keeps pushing the boundaries of what we even define as life. The early idea was that viruses just evolved from mobile genetic elements, the pieces of DNA like plasmids or transposons that escape the cell. But the discovery of these giant viruses like Mimivirus or Pandora virus, with thousands of genes unrelated to any cellular life, well, that challenges everything. It suggests they might be part of some ancient, independent lineage, completely separate from our tree of life.
That thought alone is a mind bender, but here's a final provocative thought for you to mull over, and it connects back to durability. The source mentions the incredible resilience of things like Pithovirus sibericum, a virus that was successfully revived after being frozen for 30,000 years in Siberian permafrost. So, with the accelerating pace of climate change and the thawing of these ancient environments, what other unknown viruses, or maybe even those highly durable prions, might just be waiting to be reintroduced into our modern world? Thank you for joining us for this deep dive into the world of virology.